I welcome the listeners of Radio Khanchi 90.4 FM, Hum Sabka Radio. Myself, Bharti, Assistant Professor, Postgraduate Department of Botany, Doranda College, Ranchi University. Welcome back, dear students. In this session, I will be talking about self-vaccination, which is a part of syllabus of botany at PG level, especially semester 3, and undergraduate level of semester 6. I must refer certain articles written by A.S. Berman, Theory of Centrifugation, and N.G. Anderson, that is Analytical Technique of Cell Fractions. Study of these papers will help a student a lot. Here first we'll be talking about self-vaccination. Dear students, since we know the cells are made up of large different parts, it is useful to understand each part does as well as the individual structures. In order to do this, we have to be able to study each part separately. A process called self-vaccination was developed to help with this. It takes also the part in separate the major cell organelles and other cellular structures one another. So the self-vaccination is a process of breaking down the cell for researchers or for getting different organelles inside the cell. So first, the thing comes into our mind, why self-vaccination is needed. Three main reasons. First, individual study of each cell organelle. Second, biochemical functions to be studied properly. Third, to understand the molecular basis of disease. So, if we continue the self-vaccination, we'll have to discuss first the steps of self-vaccination, which will include extraction, homogenization, and centrifugation. Homogenization is being done by grinding, high pressure, osmotic shock, and sonication. Later on, centrifugation, which is being done by differential centrifugation, that is also known as velocity sedimentation or rate zonal centrifugation. Later on, purification, that is done by equilibrium density gradient centrifugation. Let us start with the background. Since the development of the first quantitative technique for fractionating liver tissue by Albert Claude in 1946, successive improvement has been introduced by several workers that have led to reproducible methods disaggregating the tissue into a suspension of subcellular component called the tissue homogenate, and for resolving this homogenate by differential centrifugation into 4 fractions containing mainly but not exclusively the number 1 nuclei and the large cell debris. Second, mitochondria and other large granules. Third, small granules designated ribosomes, which were later found to derive largely from the endoplasmic reticulum of intact cell, as per Palade and Sickwiz in 1956, and the last, fourth one, that is soluble constituents of the tissue. So the door was thus opened wide to a new domain in cytology, the intracellular topography of biochemical functions, that is the study of the chemical composition, metabolic functions, and other properties of subcellular organelle. So, in a nutshell, we can summarize cell fractionation, which is a process for rupturing cells, separation and suspension of cell constituents in its isotonic medium in order to study their structure, chemical composition and function. Now let us begin with the steps one by one of cell fractionation. Number one, which we will be discussing, is extraction. It is the first step towards isolating any subcellular structure in order to maintain the biological activity of organelles and biomolecules that must be extracted in wild condition, called cell-free system. For this, the cells or tissue are suspended in a solution of appropriate pH and salt content, usually isotonic sucrose we are using at 0.25 mol per liter at 0 to 40 degrees Celsius. The second step, that is homogenization. The suspended cells are then disrupted by the process of homogenization, and it is usually done by four important ways. Number one, grinding. Number two, high pressure, that is also known as French pressure or nitrogen bomb. Number three, osmotic shock. And number four, sonication, that is ultrasonic vibration. So first, grinding is done by pestle and mortar or Potter homogenizer. So Potter homogenizer is very important instrument in homogenization, that a high speed blender. So homogenizer is a high speed blender that later consists of two cylinders separated by a narrow gap. So two cylinders are very important part of this homogenizer. The shearing force produced by the movement of cylinders causes cells to rupture. Second, the ultrasonic waves which are produced by piezoelectric crystals, they are transmitted to a steel rod placed in suspension containing cells. Ultrasonic waves produce vibrations which rupture the cells. The liquid containing suspension of cell organelle and ether constituent is called homogenate. So this homogenate is very important, from which further process of centrifugation and all will be done. Sugar or sucrose solution preserves the cell organelles and prevent their clumping. Third, that is the very important stage in cell fractionation, is the centrifugation. So the separation, that is fractionation, of various components of homogenate is carried out by a series of centrifugation in an instrument called preparative ultracentrifuge. So there is a big machine that is known as ultracentrifuge, in which separation of this homogenate will be done. So ultracentrifuge is also used to separate cellular components on the basis of their buoyant density, so independently of their size and shape. The sample is usually sedimented through a steep density gradient and that contains a very high concentration of sucrose or cesium chloride. So it may be known as sucrose density gradient centrifugation or cesium chloride density gradient centrifugation. Each cellular component begins to move down the gradient. So centrifugation is a process which involves the application of centrifugal force for the sedimentation of heterogeneous mixture with a centrifuge. This process is applied in industrial chemistry, biochemistry, cellular and molecular biology, as well as in environmental technology. It is also used for separation procedures such as precipitation of macromolecules, isolation of cell and subcellular organelles, and concentration of sludge. So two important terms which we'll be discussing frequently in this process, that is pellet and supernatant. So pellet is the precipitated form of particles during centrifugation, which will be in the bottom of the test tube. And another one is the supernatant, that is the remaining solution or the medium. Now, before going into detail of the centrifugation, let us understand the principle of centrifugation. So dear students, the main principle behind centrifugation is that is sedimentation of solute particle makes heavier particle sink and lighter particles to float in the solvent or medium. So the heavier particle will sink down to the bottom of the test tube and lighter particles will remain floating in the solvent or medium. The next principle, that is the solution is subjected to high angular velocity measured in RPM, that is rotation per minute, the effect of gravity, and thereby the rate of sedimentation greatly increases due to application of an external force called centrifugal force. So this is very important upon which the centrifugation process continues. Dear students, for this centrifugation process there is a device known as centrifuge which are used for separating particles from a solution according to their size, shape, density, viscosity of the medium and rotary speed. So the centrifuge spins the suspensions around a central axis, producing centrifugal force which causes the suspended particle to collect as a pellet in the tube bottom. Rest fluid containing suspended particulate matter called supernatant. So there are few important parts of a centrifuge, that is core part which is a rotor, second fixed number of arms radiating from it a certain angle, and third low tube attached for sample placement, they are the hollow tubes, and fourth a big ball bearing, and fifth aluminum titanium graded drum and motor and brake assembly, controlling knobs and big basket and all. So these are the important parts of a centrifuge. Now during centrifugation process, the homogeneate or homogenized suspension is placed in a centrifuge and spun at a high rate of speed, and this process is called centrifugation. And that speed that can be varied, so we called differential centrifugation. During centrifugation, different part of the cell will accumulate in the bottom of test tube in a pellet. Reason for this is because the different part of the cells are bigger or smaller, and as bigger they are, quicker they will sink to the bottom, and you don't have to spin it as fast. Whereas the other pieces are smaller, you have to spin it at a very higher speed, that forcing them to settle at bottom of the test tube. So for example, if we first spin the tube at something like at 100 G, that is 100 times the force of the gravity for 10 minutes, you can get a pellet rich in nuclei and cellular debris at the bottom of test tube. But there will still be a liquid portion above the pellet called the supernatant, and within the supernatant there will be other parts of the cell that are smaller. So if you pour the supernatant off and spin in a higher rate of speed at something like that 20,000 G, you will get a pellet rich in mitochondria. If you repeat that process and spin that supernatant down at something like 80,000 G, the pellet will be rich in microsomes, that is pieces of plasma membranes and cells internal membranes. So if you have spin the supernatant down again at 1,50,000 G, you will get a pellet rich in ribosomes within 3 hours. So this technique allows the isolation in subsequent research subcellular components, so you can determine more about their individual role in the cell. Now, after the centrifugation process is over, the process moves for the purification that will be done by density gradient centrifugation, which is a procedure of separating particles such as viruses or ribosomes or molecules such as DNA, in which the sample is placed on a performed gradient such as sucrose or cesium chloride. So accordingly, they are also known as sucrose density gradient centrifugation or cesium chloride density gradient centrifugation. Upon centrifugation, either by rate journal or equilibrium procedures, the macromolecules are banded in the gradient and can be collected as a pure fraction. So this density gradient centrifugation may be done by rate journal centrifugation or isopycnic centrifugation. The organelle fractions, that is the pellet, obtained in the differential gradient centrifugation is purified by equilibrium density gradient centrifugation. In this method, the organelles are separated by their density, not by their size. So the impure organelle fraction is layered on the top of gradient solution, for example sucrose solution or glycerol solution. The solution is more concentrated at the bottom of centrifuged tube and decreases in concentration gradually towards the top. So in the gradation tube, the concentration of sucrose solution is very higher at the bottom, and as you will move towards the top, the gradient solution there will be decreasing in their concentration. So the tube, when centrifuged at high speed, the various organelles migrate to an equilibrium position where their density is equal to the density of the medium. So they are purified accordingly to their density, which is matching to the density of the medium. And dear students, if you remember, Messelson and Stahl and Vinograd in 1957, they used denser cesium chloride CaCl2 gradient for separation of heavy DNA with nitrogen 15 and from the DNA with nitrogen 14 to provide evidence for semi-conservative nature of DNA replication. I hope you remember this experiment of Messelson and Stahl. In conclusion, we say that one can learn about cells depends on the tool at one's disposal, and in fact, major advances in the cell biology have frequently taken place with the introduction of new tools and techniques to study the cell. Thus, to gain difficult types of information regarding cell biologists have developed and employed various instruments and techniques. A basic knowledge of some of these methods is earnestly required. Dear students, so these were all about cell fractionation, and which is being done by the different processes like their extraction, homogenization, and centrifugation. After that, purification for assessment and all. So I hope you have got understanding over the different things. Rest part related with this centrifugation, like cesium chloride density gradients centrifugation, sucrose density gradients centrifugation, marker enzymes and all will be discussed by me later on in the next part of this lecture. So I hope you all have enjoyed this talk. Thank you very much. Thank you for listening. Jai Hind.